Okay, adding on to our genetics, we're going to look at dihybrid problems again, looking at two different genes at once. When we did dihybrids before, we assumed that the genes were on different chromosomes. So unlinked, that's what we call unlinked. They're not on the same chromosome, so they're different chromosome pairs. So let's go through this. We're going to look at two traits at once. The color of the fly's body. So we're piggybacking off on Morgan and his fruit flies. And this is something that I said that he figured out that certain genes were on certain chromosomes, and that all the genes on that same chromosome are linked together. And so we have been talking about unlinked. So we got the gray and black body. So that's the trait, is the body color. And here's the B plus. The plus, remember, means wild type, which means most flies have a gray body. I'm going to tell us here that gray body is dominant, because so it doesn't tell you what's dominant or recessive. So like I said, I like to sometimes use that big B. So it'd be big B plus. All right, you could use that as well. All right, and then the normal versus vestigial wings. He used VG again, looking at the non-wild type, the mutant phenotype. So he used VG for vestigial. That's why I used B over here for black for the mutant. So this one is dominant. So it happens to be that the wild type in this case here is dominant. And so you could use, if you wanted to, if it's confusing for you, VG plus with a capital V, just to reiterate the difference between dominant and recessive. But for this one, the wild type happens to be dominant, and so we could get away with just using the pluses. All right, so let's look. I'm gonna do some color coding. So we're gonna look at if they're on different chromosome pairs. So that means that, let's say the gray and black body gene is on chromosome pair number 1. All right, and so looking at this individual, this is our cross. This is the first individual. They are B plus, which means they have a gray body. VG plus, which means they have a normal body, so they're gray and normal. So I'm going to write that, gray, normal wings. All right, cross with BB, which is black, and VG, which is vestigial. So that's what we're crossing. Notice that these guys have opposite traits, gray and normal, black and vestigial, and they're both homozygous for their traits. All right, so so on the chromosome pair number 1, you have the B plus gene and the B plus gene, and then you have, here's my new color, chromosome pair number 2, and you have the wing gene. So let's say it's right here, and this one is VG plus. VG plus. All right, so then we're crossing that. All right, so let me just use my black pen. Crossing that with this parent here. This parent on chromosome pair number 1 has the 2 B genes, no plus, and they also have a chromosome pair number 2 with the VG genes. This is my cross. All right, so remember, when you cross them, when you do a Punnett square, this is like what we did before. They're going to give one chromosome pair number 1 gene, one chromosome pair number 2 gene. All right, so this one could pass on a B plus with a VG plus, right? A B plus with a VG plus, B plus with a VG plus, and a B plus with a VG plus. So remember, when you do the Punnett square, we can do a big Punnett square where then you have the 4, this, you have the 4 like this, all right? And that parent would be B plus, VG plus, B plus, VG plus, B plus, VG plus, B plus, VG plus. Do you see that these are all going to be repeats? All right, so you could just do it one time. All right, so you could do that. And then what's this parent going to be? They could pass on this B with this VG, this B with this VG, this B with this VG, this B with this VG. So they're all going to be B and VG. All right, so I don't have to. You can write it 4 times. You don't have to, though. So really, it's a 1 by 1. They're really B plus B, VG plus VG. That make my box big enough there. All right, so they're all heterozygous. 100% of the kids are all heterozygous. What phenotypes would they have? They would be gray body, because B plus is dominant to B, and they'd be normal wings, because VG plus is dominant to vestigial wings. All right, so the kids of that cross are all going to be B plus B, VG plus VG. All right, so now what we're going to do is draw what their chromosomes look like. So the B plus B, B, all right, I should have, I'm going to go over that in my color, because that's this gene. So why they have B plus is because this parent gave them a chromosome pair number 1 that has a B plus on it. And this one gave them a chromosome number 1 with a B on it. Hence the B plus and the B during meiosis. They split, remember, and they only give 1 of each of the chromosomes. Then the VG plus, oops, let me put my cap on that. The VG plus, VG, I'm going to go over that right here. Chromosome pair number 2, VG plus VG. And so that's why they look like that. All right. So then next it says, do a test cross with the offspring of the first cross. All right, that's this. And figure out the phenotypic ratio you would expect. All right, so let's look at this. So if we do a cross, so this is the kid of the first cross. So we're going to do a test cross. All right. Do a test cross. What is that? We talked about that in our previous notes. Test cross here reveals the genetic makeup of an organism with the dominant trait. And so this is when we take the dominant individual with the dominant trait and cross it with a recessive individual. All right, so let's look at this. So then the cross is going to be crossing it with all recessives. So recessive is the black body and the vestigial wing. So that we're going to cross it with a little b. So what is their VG genes are going to be VG, all recessive. So what are their chromosomes going to look like? So they also have a chromosome pair number 1. They're going to have a little b on those. And they're going to have a chromosome pair number 2. They're going to have a VG on each. Okay, so this is our cross. So now let's do the Punnett square, all right? And we could write that here if you don't remember when you're looking at this later. This means cross with an all recessive individual. Okay. So what are our boxes going to look like? So let's look at parent number 1. They could pass on the B plus and the VG plus. I'm going to go ahead and write that here. B plus and VG plus. They could pass on the B plus and the VG. They could pass on the B and the VG plus. Or they could pass on the little b and the VG. All right, so that's our top of our Punnett square. So we do have 4 possibilities, so we can't reduce it like we did up here because there's no repeats here. All right, so we have our 4 possibilities, and then here, look at here, they're going to either pass on this B and this VG, this B and this VG, this B and this VG, or this B and this VG. So the only thing, I'm not drawing my Punnett square yet because I want to see how big I had to make it. I'm only going to write it 1 time. You could write it 4 times to have a 4 by 4 Punnett square. I'm only going to write it 1 time because 100% of their gametes have a B and a VG. So now I'm going to make my Punnett square like this, where these are my 4. All right here. So the first kid could be B plus B, VG plus VG. Second kid, second type of kid, B plus B, VG, VG. Next kid, B, VG plus, forgot what I was doing here, VG plus, VG. And lastly, B, VG, VG. So notice here, then what I want you to do, if I were you, I would pause this and do the phenotypic ratio. All right, so what ratio would you get? What would these guys look like, just for practice on that? Okay, so hopefully you paused that and did that. You should have gotten this one. We get 1 gray in normal wings to this one, 1 gray vestigial wings. To this one here, 1 black-bodied normal wings. To 1 all recessive black and vestigial wings. So we get a 1 to 1 to 1 to 1 ratio. All right, so that's what you would expect if they're on different chromosome pairs. All right, so let's look at this new thing. What if they're linked? This is like our meiosis picture. It says now doing a cross assuming that the genes are linked. Linked genes are 2 genes that are on the same chromosome pair. So we assume that they were on 2 different pairs here. Now we're going to make it on the same pair. So now let's say both of these genes are on chromosome pair number 1. So here's chromosome pair number 1. And so they're this parent is B plus, and then the wing genes are on the same chromosome. So let's say it's down here. They're on the same chromosome instead of different. Okay, so now I'm going to do the different colors by the different parents. So then we're going to cross this parent. So that's the black and normal wing, or sorry, gray and normal winged fruit fly. And then this is going to be all recessive. So they're going to have little b, and then for their wings, they're going to have VG and VG. This is the cross. All right, so now let's look at how different that Punnett square would be than it was on the opposite page when we were talking about it being unlinked. So now, do you see that these these genes that are on the same chromosome, they get inherited together. When this parent makes their egg and sperm cells, they give the B plus and the VG plus together because they're on the same chromosome. B plus and VG plus together. So therefore, there's only one kind of egg or sperm cell. They're going to give this chromosome or this chromosome, but both of them have a B and a VG. I set it up like that because there's only 2 possibilities there. But you could also look at this and go, no matter which chromosome they give them, it's going to be 100% the same. So you could do a 1 by 1. So all their kids are going to be B plus B, VG plus VG. So they're heterozygous. This is like this here, all right, that we got over here. All the kids are B plus B, VG plus VG. That's the kids of that cross. Let's look at what their chromosome number 1 looks like. So then what does their chromosome number 1 look like, that pair? They got one of these 2 chromosome number 1s from mom and one from dad. So the one that they got from mom has to have a B plus and a VG plus on it. And then the one that they get from dad also has to have a B and a VG. And you know what? I want to I just realized to show what I want to show next. Oh, this will be fine. All right. So now we're going to do the same thing that we did before. All right, is cross that with a homozygous recessive. We're going to do a test cross here. So now this is where I'm going to use my second color. All right, so we're going to cross it with the test cross here. All right, we're going to cross it with all recessive like this. If you're all recessive, what does your chromosome pair number 1 looks like? So then I'm going to do a blue here. All right, here. So here's this one's chromosome pair number 1. They're going to have B, B, VG, VG. All right, so that's what that's going to look like. So what kind of kids would you expect with this cross? So let's do this cross. This parent's either going to pass on this chromosome pair or this, right? This or this. And so they're going to pass on the B plus VG plus together, or the B and the VG. Those are my two combinations here. I guess I could have done it to relate this, that these, let's go over that. I should have done that. To relate those to that parent. All right, those are the possibilities for that parent. And then this parent's possibilities is this B and VG chromosome or this B and VG. So either way, they are giving a B and a VG. Okay, so let's do our Punnett square here. So what would you expect? All right, I'm not going to color code. I just wanted to show that these two are giving, these are those two parents here. So they're B plus B, VG plus VG, and B, B, VG, VG. So we would expect in our phenotype ratio 2 different kinds of flies. It's going to be a 1-to-1 ratio. When you're looking at this, they're going to have the dominants, all right, the 2 dominant traits. So B plus was, if we look over there, that was gray, right? So 1 gray bodied. And the VG plus, which was normal wings. 2, this one here, which is 1 black vestigial. Okay, so that's what you would expect. So in that, so do you see here that if you do the same crosses, all right, the parents have the same genotypes on either side here, all right? So you're crossing them. You, in both situations, your kids, when you cross the homozygous dominant, homozygous recessive, you get heterozygous in both linked and unlinked. So how do you tell then, if you want to go, okay, if these genes are linked or unlinked, then you do one step further and you do a test cross. And the reason why that's going to be helpful is that you get different ratios. So you're going to get a 1-to-1-to-1-to-1 ratio, and you're going to get you're going to get a 1-to-1 ratio. So here you have different combinations that you don't have here if they're linked. So that's your first clue if the genes, the 2 genes you're looking at are linked or unlinked. All right, now if we look here, Morgan, when he did this, this is what he got when he actually did this cross. We just said you'd expect a 1-to-1 ratio. He did not get a 1-to-1 ratio. So the gray normal, here's those guys, black vestigial, those look like a 1-to-1 ratio. They have about the same numbers of each in those, but then you get the 2 other possible combinations that we didn't get weren't able to get. He got the ones that are over here, the other 2 that we didn't get. But he got them in way lower numbers, all right, than what he'd expect. So so it says, can you explain his results? So thinking back on meiosis and doing the meiosis drawing, what happens during meiosis that can switch up genes? What happens is crossing over. So I want to show you here what happens when crossing over occurs. And so when looking at this, these 2 here, let's look at if crossing over occurred here. To do that, let's see here. To do that, I'm going to have to, I have to go over this and make this a different color so we can see. So I'm going to go over this and make this one orange. All right, just to make it a little bit different. So let's say that these guys, this isn't a cell, and this cell is going to go through meiosis. So the first step in meiosis is to replicate the DNA. So let's do that. So you're going to have, let's do this. This half is an exact copy of this half, and we're going to show crossing over. And I'm going to replicate this that I made orange here. I'm going to do the crossing over like this. Here's the, all right, remember that? All right, so let's put the genes on here. So this one has the B+ genes and the VG+. All right, now notice here the VG+ is after where they cross over. That's important because they're going to switch genes. Then the orange chromosome has the B's, B, VG, and VG on that one. Okay, so fast forward, they line up during meiosis, they line up together with their partners. You get the spindle fibers that are going to pull them to opposite sides of the cell. The cell is going to indent, and we're going to go through meiosis 1. I kind of have to make these small cells right here. Meiosis 1, where these two chromosomes, the pink and the orange, get pulled to opposite sides of the cell. So let's look at what this one's going to look like. The replicated chromosome here is going to have once they get pulled apart, they switch from the point of crossover down. So this is orange. And then the orange chromosome is going to have the pink in the end, like this. So and what's going to be on that the pink is going to have the VG gene. And then let's do the pink over here. Since I've got the pink, this is going to be B+, looking up here, VG+. But instead of the VG+ over here, it's going to have the VG. And this one's going to have the B, the VG. I forgot to put the plus, didn't I, over here? I think I got. All right, so that's the VG+. All right, so now it's got a new combination. So then let's go ahead and fast forward through meiosis. Spindle fiber attaches to both sides. Spindle fiber attaches to both sides. And meiosis 2 is going to pull the chromatids to opposite cells. So you're going to end up with 4 cells. So I'm just going to do kind of 4 cells like this. This is meiosis 2. And what's going to be in this one? This single piece of DNA now, it unravels into chromatin that has a B+ and a VG+. Over here, you're going to have the one with the orange end. So that orange end is going to have a VG, and the pink is going to have a B+. Here, you've got the orange. Let me switch colors here. You've got the orange with the pink end. That's VG+. And then the orange is B. And then this one's all orange, no crossing over occurs, so it's going to be the B and the VG. So now look at when crossing over occurs. Before crossing over occurred, you only had 2 combinations. All right, they could pass on the B+ and the VG+, or the B and the VG. All right, but now that crossing over occurs, now you've got 2 new combinations here. I'm going to put that in orange. These guys right here, or sorry, did I say orange, green? These guys are new combinations due to crossing over. Due to crossing over. So these were the original right here. no crossing over occurs. These are the new ones. So now let's add this. So then realistically, if crossing over occurs, they could have these two new combinations. So they could have the B plus and the VG, or they could pass on the B and the VG plus. And so let's add on to this. So in green are the new combinations. So if crossing over occurs, then you could have a B plus B plus B meet, or VG, VG. Or you could have a B, B, VG plus VG. And so what happens is these are those two new combinations. This one would be gray and vestigial wings. And look at here, that's this one here, gray and vestigial wings. Got a little bit, 206. And then this one would be black and normal wings. That's these guys here. So these guys right here, are new due to crossing over. All right, so they're new due to crossing over. And why they're in such lower amounts, why it's not a 1-to-1 to 1-to-1 ratio, is because crossing over doesn't occur 100% of the time between those two genes. So sometimes it does, sometimes it doesn't. So more often than not, it doesn't. So therefore you get most of the kids being what you would expect with no crossing over. But then sometimes when crossing over occurs, you can get these two kids. And so he got some kids like this, all right, when he was doing his experimental fly crosses. We call these new ones that you don't, that are due to crossing over, we call these recombinants. Think of them as they're due to recombination, all right? So recombinants due to crossing over. All right, and then these guys here, these guys are called the parentals. Why are they parentals? Why are they called parentals? All right, because if you look, gray and normal, black and vestigial, these guys, they're like their parents, gray and normal, black and vestigial. So that's why they have the same phenotype as their parents in that 1-to-1 ratio. The new ones, the recombinants, are the ones that don't and are due to crossing over and recombined. All right. And so what you can do is, again, if you don't know if genes are linked or not, you go through this step. You take a parent like this and a parent like this to get heterozygous, and then you take the heterozygous and you you cross it with a homozygous recessive, and you look what kind of ratio you get. If you get all four, but you get overwhelmingly like the parents and only some like that, and it's not a true 1-to-1 to 1-to-1 ratio, which is what Morgan got, that's what tells you it's linked. If you do get a true almost 1-to-1 to 1-to-1 ratio, then it's unlinked. All right. And so what we do is one step further is when we do experiments like this, we can figure out the percent of crossing over between the genes. All right. So when we look at that, what we do is go, okay, out of all the kids, how many of them are due to crossing over? So what you do is you take 206 plus 185, which will give you 391, divided by how many total kids that they had. So 965 plus 944 plus 206 plus 185. I already did this. It's 2,300. And so then you divide that. When you do that, if we round to 2 decimal places, you get to 0.17. You multiply by 100 to get a percentage, and the crossing over frequency between those two genes is about 17%. You just can figure that out based upon how many kids are due to crossing over out of the total. Okay, and so when it says, can you explain the results, what I would like you to do is write into words what I just explained, all right? And then you're going to turn this in, and I'm going to check your writing in words that you understand how he got these guys, okay? All right, turn this into Google Classroom.